Bismillahirrahmanirrahim. Dear fellows, Assalamu alaikum. I am Dr. Sabir from the Department of Environmental Sciences and Engineering, Government College University, Faisalabad. Today I am here for an oral presentation in the third International Conference on Applied Zoology. The topic of my presentation is biodegradation of isoproturon by a bacterium Sphingobium species S29 in soil and aqueous media. As you know, that pesticides are used in conventional agriculture to ensure the quality and quantity of agricultural yield. These pesticides might be in the form of fungicides, herbicides, and insecticides. Although the pesticides are used for the improvement of agricultural yield, but at the same time, they are source of pollution of the environment. Isoproturon is one of the phenylurea herbicides which has been commercialized for more than 50 years. It is used for pre and post-emergence control of broadleaf weeds in cotton, fruit, cereal cultures, and other agricultural crops. It is a systemic herbicide which inhibits the photosynthesis of the target weeds. Ecotoxicological data suggest that it is toxic for some invertebrates. It is suspected to be carcinogenic and genotoxic. It causes reproductive and developmental toxicity in human beings. In addition to this, it has been detected in waters in several countries, including Pakistan. After entry into the soil, few previous studies have reported for a very low mineralization of this herbicide in the soils. However, during the recent years, some of the studies have reported for enhanced degradation of this herbicide in the soils repeatedly treated with this herbicide. This phenomena is attributed towards the adaptation of microorganisms for isoprotein degradation. The microorganisms use this isoprotein as source of their energy. This phenomena has been observed in different countries including UK, Denmark, France, China, and Pakistan. On the basis of these results, several researchers have isolated different bacterial and fungal strains involved in the biodegradation of isoprotein. Here are a few strains of bacteria and fungi which have been isolated and characterized for biodegradation of isoprotein. These are the strains which have the potential for mineralization of this herbicide. In this context, the present study has been planned with the objective of isolation and characterization of a novel bacterial strain involved in isoprotein degradation in aqueous as well as soil media. For this bacterial strain was isolated through an enrichment culture technique in the minosalt medium using isoprotein as only source of carbon. This strain was identified through PCR amplification, sequencing, and bioinformatic analysis of its 16S ribosomal DNA gene. The potential of the strain S29 for degradation of isoprotein as well as its known metabolites was tested in minosalt medium. The potential of this strain to degrade other phenylurea herbicides was also tested in minosalt medium, and the capabilities of this strain to degrade isoprotein in the mineral salt medium was also tested at different temperatures and pH values. And finally, this strain was tested for degradation of isoprotein in the soil. It was found that this strain had a good potential for degradation of isoprotein as well as its known metabolites as shown in this figure. On the basis of this finding, it was suggested that this strain followed a degradation pathway in which the isoproteron was demethylated twice, and then ultimately it was transferred into 4-isopropyl aniline. This pathway has already been reported for some other bacterial strains belonging to genus Sphingomonas. This strain had the potential for only further degradation of isoproteron, and none of the other members of phenylurea herbicide was found to be degraded by this strain. It means that this strain has the enzymatic system which is highly specific for degradation of isoproteron. The optimum temperature for degradation of isoproteron by this strain was observed to be 30 degrees centigrade, and on other temperatures the potential was found to be reduced. The optimum pH for isoproteron degradation by this strain was found to be 7, whereas it was decreasing with any increase or decrease in the pH values. And furthermore, it was observed that degradation at different pH values is also associated with the growth of the strain at different pH values. In the soil, it was found to have a good potential for degradation of isoproteron. This study indicated that in the soils by augmented with the strain S21, S29, whole of the isoproteron was degraded only within 20 days. Hence, this strain has a good potential for the degradation of isoproteron in aqueous as well as soil media. It can be concluded that this strain might be used as a potential candidate for the bioremediation of the soils as well as aqueous media contaminated with herbicide isoproturon. Thank you very much for your attention.